For today, we will start with the presentation by Pravina about Maxwell Biosystems and by Scott about the work that was done with Fujifilm Cellular Dynamics. So now I'll transfer to Pravina for the next presentation.
Great, thank you very much for the kind introduction. So Maxwell Biosystems is a global technology leader in high-content electrophysiology. And from our headquarters here in Zurich, our offices and labs serve as a hub for a global network, with our system hosted in laboratories across 23 countries around the world. Backed by more than 20 years of R&D and a strong reputation in the scientific community, we provide cutting-edge, high-density MEA hardware, software, and applications that has enabled users to publish in nearly 200 peer-reviewed papers in leading scientific journals. Here we have a few recent examples of our user publications in high-impact journals, and for a more complete list, please visit our website or scan the QR code here. So using our high-density MEA platform, researchers have made significant advancements in the study of cells and disease modeling. For example, Dr. Marian Roschka-Plokan, in his postdoc at the Polymenduo lab, investigated neurodegenerative diseases such as ALS and FTD, where he developed a new iPSC-derived human neuronal network model called iNets. And in collaboration with ETH Zurich, his Nature publication featured the functional characterization of this innovative neuronal model using our single-well MaxTwo HD-MEA platform. Another example is research conducted by Dr. Maria Sundberg's team at Harvard Medical School, where she moved from our single-well MaxTwo platform to our multi-well MaxTwo system, eventually leading to the discovery of hidden phenotypes associated with neuropsychiatric disorders. And this highlights how our system empowers our users in their efforts to develop treatments for different brain disorders. So how can you achieve this? Well, at Maxwell Biosystems, we believe that every cell has a story to tell, and for that, you need high-resolution readouts. While traditional methods such as imaging techniques can be very useful for studying neuronal morphology, these often involve dyes that can influence cell behavior. Alternatively, while patch clamp is a powerful technique and a gold standard for examining single-cell function, its low throughput limits the scope of analysis. So at Maxwell Biosystems, we develop next-generation electrophysiology platforms with high-density microelectrode arrays that bridges these two approaches together, enabling high-quality data collection from living cells without invasive techniques. And what insights can you gain using MEA technology? Well, imagine you have cultured neurons in an MEA chip. So with this technique, you can either record spiking activity to capture firing rates and action potentials, or visualize activity maps here to see where and how cells, how active neurons are. And you can also explore network dynamics using raster plots to track synchronized patterns. So with such MEA devices, you can achieve powerful insights, but success depends on a few key elements. And reliable, high-quality results are essential for ensuring meaningful and impactful research. So Maxwell Biosystems solutions are designed to support you by providing comprehensive neuronal characterization for deep insights into network behavior, reproducible results under optimized conditions, simple and seamless workflows for everyday use, and advanced capabilities to push the boundaries of neuroscience research. Neuronal cultures contain diverse activity profiles, so capturing signal from as many neurons as possible is key to fully understanding the complexity of your network and obtaining reliable results. So low-density MEAs are often used, but can miss activity from neurons too far away from electrodes. While with Maxwell's HD-MEA solution, we have 26,400 electrodes per well at a pitch of 17.5 micron pitch. And this ensures that every active neuron is recorded. Furthermore, if you work with iPSC-derived neurons or organoids, you're likely aware that action potential signals can be very small, especially early in development. And even when using high-density MEAs, capturing such small signals isn't always guaranteed, especially if the system suffers from high background noise, which can easily be masking small neuronal signals. With Maxwell's HD-MEA platform, we offer very low noise readouts and ultra-high dense electrode coverage, achieving greater fidelity than other systems for reliable single-cell resolution recordings of even the smallest signals. Our axon tracking assay harnesses this by uniquely enabling you to track small action potentials at subcellular resolution, reconstructing axonal morphology from electrical activity and measuring propagation velocity, all label-free and non-invasively. Overall, our HD-MEA technology features a large sensing area of 2 by 4 millimeters square, allowing for dense and highly detailed recordings. We also have 26,400 electrodes per well and delivering 3,265 electrodes per millimeter square. You also benefit from low noise readouts, flexible stimulation with up to 32 channels simultaneously. And what really sets our technology apart is scalability without compromise. So even in 6 and 24 well MaxTwo plates, each well retains the full electrode density, enabling higher throughput experiments without loss of signal quality. Maxwell Biosystems offers 2 HD-MEA platforms to meet diverse research needs. So we have the MaxOne system, which is a flexible single-well system ideal for advanced applications requiring fine control and customization, including integration with other technologies. While our MaxTwo is a high-throughput platform for 6 to 24 well plate options, enabling scalable long-term recordings without compromising electrode density or signal. We also offer the MaxLab Live Suite, which is an all-in-one software solution for high-resolution electrophysiology to not only visualize, record, analyze, and produce metrics and reports of all your data. The software includes several assays enabling comprehensive functional characterization of cultures. This includes the activity scan for label-free mapping of active cells and activity distribution, network assay for bursting and synchronization of neuronal populations, axon tracking assay for subcellular analysis of action potentials and neuronal footprints, and the stimulation assay to evoke neuronal activity and excitability profiling. And together, these assays provide a complete functional profile of your culture. Overall, our technology enables the characterization of a wide range of samples, including 2D primary neurons, iPSC-derived neuronal cultures, as well as 3D models such as brain organoids and spheroids. And our platform can also be utilized for acute retina or brain slice applications, or even advanced neurocomputing or microphysiological systems. So whatever your sample of interest, don't hesitate to contact us. We're eager to help you find the best solution for your functionalization. So furthermore, our system can actually capture functional activity across a wide range of 2D neuronal cultures, from fast-bursting primary neuronal cultures to low-amplitude dopaminergic neurons that are typically hard to detect. And accurate disease modeling of 2D neuronal cultures is also possible thanks to high reproducibility across wells and experimental conditions, enabling reliable differentiation of pathological features and robust insights into disease-related dysfunction. For example, in isogenic ALS models with eye-cell spinal motor neurons carrying TDP43 mutation, we observed consistent sparse network activity and altered dynamics compared to healthy control lines under different conditions. And building on that, I would like to highlight our new application note with FUJIFILM Cellular Dynamics, which showcases the MaxTwo 24 well plate system for longitudinal functional profiling of frontotemporal dementia model using eye-cell-induced excitatory neurons. And you can read more here by scanning this QR code as well. And with that, I'm pleased to introduce Dr. Scott Schachtel from FUJIFILM Cellular Dynamics, USA. He will be presenting on the application note and more in his talk on revolutionizing neurodegenerative research, case studies, and innovation for HD-MEA assays with human iPSC-derived neurons. Thank you.
You so much for the invite to come in and share some of the research you've been doing at FUJIFILM Cellular Dynamics and in collaboration with MaxTwo currently and over the years. And so as you can see from the title of my talk, I'll be focusing on models of neurodegenerative disease. So to start, just give a little back history on FUJIFILM. So FUJIFILM has a history of innovation starting with film and then more recently moving into healthcare, where we've acquired a portfolio of complementary life science companies to support preclinical research and discovery. FUJIFILM Cellular Dynamics, or as what I'll refer to as FCDI throughout the talk, is a part of this portfolio, and with it brings innovation in induced pluripotent stem cell technologies, as well as industry-leading iPSC-derived cell manufacturing. So to elaborate slightly further, FCDI has 4 core competencies, which are underscored by our proprietary iPSC reprogramming technology and licenses, which fuel, due to the replicative ability of iPSCs to provide a renewable source of starting material, which then can be differentiated into different cell types. And so similarly, we have proprietary differentiation protocols that facilitate the industry scale manufacturing of a large variety of iPSC-derived cell types. In fact, we're one of the oldest iPSC-based cell companies and therefore have unparalleled QA, quality control and quality assurance around our products. So all of these core competencies fuel the discovery and cellular therapeutics business within FCDI, which is exemplified in this diagram cartoon on the screen here. But for the focus of today's talk, as it relates to today, we'll be focusing on our research products that can enable drug discovery and disease modeling. So the top half of this picture. And on the discovery side, we manufacture a large variety of iPSC-derived products. They're delivered as cryopreserved vials alongside typically a medium for culturing. And as you can see on the upper left-hand corner, the largest portfolio is the neural portfolio. And this slide is just depicting the offering we have featuring cells differentiated from healthy donors. And later in the talk, we'll get into some of the disease models that we also offer. But that's just in a little bit. And first, I wanted to quickly illustrate the quality of our products and the consistency. So this slide contains purity data for 3 of our iCell neural products, GABA neurons, dopaneurons, and motor neurons. And as you can see, for each product, the purity is quite high, speaking to the specificity of our differentiation protocols. And the error bars are quite narrow, reflecting the precision and consistency of our manufacturing and our products. And so this quality really translates into a reliable source of cells, human-derived cells, for the research community to utilize so they can not worry about the differentiation process, but strictly focus on the research questions they want to ask. And across our neural portfolio, our cells can be cultured in a multitude of combinations, from monoculture, shown on the right, to various degrees of co-cultures, tricultures, and within 2D or 3D systems. And so we're invested in a broad characterization of our cell types. And as you can see below each of the images, various types of applications and assays that we've typically used within either monoculture all the way through co-cultures. And so these assays are typically used to help validate the function, but also provide a means to help us and the user establish a baseline to compare against when they're looking at disease-inducing conditions or cellular disease models. And so for the purposes of today's talk, we'll obviously focus on MEA. And we'll move into that kind of background of the history of MEA with an FCDI and Maxwell collaborations. So as MEA was introduced into the field, it became really a key technology for FCDI to characterize our cells and the ability to record field potentials from a neural population and monitor the development of network activity of our cells over time in a non-invasive manner. This was a perfect solution for this. So we've historically used this tool using traditional MEA platforms to characterize our neural products, starting with our eye-cell GABA neurons, which are featured in the upper left-hand corner, and then even more recently into our recent products like our eye-cell sensory neurons in the bottom right-hand corner. And this testing and characterization often results in application notes or lab protocols that are featured on our website and can be downloadable for users who are interested. And with these traditional lower resolution MEA platforms, for our initial characterization studies, these were really very helpful, and we learned quite a bit in how to utilize our cells on an MEA platform. For example, on the left, looking at how excitatory and inhibitory ratios of neurons can impact network burst firing rates. And on the right, how the addition of astrocytes, iPSC-derived astrocytes into the culture can promote earlier and better organization of the network activity. So this technology has been quite informative for us. Now, however, as our portfolio has expanded into disease models and more complex culture setups like co-culture and triculture, we quickly learned that the traditional methods had limited spatial resolution, and the number of electrodes did not provide the capacity for us to do recording from targeted individual neurons and large networks to really fully understand and evaluate the capacity of and differences in disease phenotypes across our different models. And so the solution for this was really the high definition MEA. And so it was very exciting for us when Maxwell Biosystems released their HD-MEA platform. And we were happily an early user and collaborator with them, starting back in 2017, and then publishing an application protocol, the first one, collaboration between us, nearly 5 years ago in 2020. This is shown bits of it, the protocol, the setup of using our cells in the Maxwell system here on the screen, using the MaxTwo chips, and describing the preparation of gluten neurons and astrocytes in co-culture, as well as in the application note describing how to utilize some of the MaxLab live assays that are offered in that platform. And so just to give you a little taste of what that data looks like, we can see very nice, fantastic looking data develop over time. On the top, we see longitudinal analysis of network firing and synchronized bursting over time. And then on the bottom, we could nicely see consistent results across different MaxTwo chips produced, which was really important for us because it gave us a great confidence both in the platform and the ability to start branching out and exploring more features of the software in the system and across different cell types that we offer. So in particular, we're very interested, as I've kind of already mentioned, in MEA profiling of neurons within neurodegenerative diseases or carrying neurodegenerative disease associated mutations. So just for a brief background of our portfolio here, this lists our off-the-shelf portfolio of disease models. And as you can see, our portfolio focuses on 4 main categories: Parkinson's disease, Alzheimer's disease, ALS, FTD, and then neurodevelopmental disorders, more specifically focused on epilepsy at this time. So we have a range of options from healthy cells engineered to express risk-associated mutations in healthy patient lines, and then also actually taking patient material from people diagnosed with these diseases and differentiate them into our specific cell types, and these patients do carry risk-associated genotypes in them. So a combination, and you know, an important piece of this is that we try to make sure that all of our disease lines have healthy and isogenic mutation-corrected controls to compare against to accurately look and define those disease phenotypes. So as our disease panel began to grow, we wanted the ability to assay in one plate multiple genotypes and do so across replicate wells. And this was really enabled when Maxwell Biosystems introduced their MaxTwo 24 well plate HD-MEA system, which provides 24 wells of high resolution recording capacity, as Pravina already nicely laid out for us at the beginning of the talk. So for the rest of my talk, I wanted to go through a few case studies of how we've modeled neurodegenerative disease on the MaxTwo platform to compare across disease lines. And a lot of this work was done actually in collaboration with Maxwell Biosystems. All right, so to start, the first case study is looking at ALS and FTD using HD-MEA and focusing on some of the data we generated in the most recent application note, again, that Pravina nicely shared at the beginning of the webinar. So frontotemporal dementia and amyotrophic lateral sclerosis, ALS, are on a disease spectrum with common underlying gene mutations that can lead to motor neuron disease, cortical degeneration, and other associated memory or behavioral changes. And so within this spectrum, mutations in the progranulin gene are a predominant risk factor for frontotemporal dementia, accounting for up to 20% of familial FTD and 5% of sporadic FTD. So specifically, granulin mutations such as the R493X missense mutation result in a haploinsufficiency of progranulin in the brain. And some of the implications for granulin deficiency are shown on the right, which include protein mislocalization, lysosomal dysfunction, neuronal and synaptic loss. So as I mentioned earlier, formation of neural networks on multi-electrode array is a great way to monitor neural activity, and we want to use this to identify disease phenotypes. This slide shows some background data on previously published literature on a few papers that have looked at the FTD phenotype in granulin R493X mutations on MEA activity with mixed results. So on the left, we see that in this study, we see potentially increased activity, little to no activity on the left, and then potentially increased activity on the right. Now, both of these studies here use iPSC-derived neurons with a homozygous knockout for the granulin mutation, which has been traditionally used, and was used in these because the mouse models require this homozygous knockout to see a phenotype. And it's been hard to detect a phenotype using a heterozygous knockout for the R493X mutation. However, we sought to model a heterozygous haploinsufficiency of granulin using the R493X mutation. And we wanted to utilize HD-MEA with that model to see if we could detect a phenotype. And so in order to do this, we utilized our Neurodegenerative or NGN2 forward-programmed I-cell-induced excitatory neurons. And so using this differentiation method, we can produce a highly pure population of general excitatory glutamatergic neurons. You can see this in the data on the bottom, where we see nice high purity of beta-3 tubulin in the population. And on the very right, looking at a gene expression profile, seeing very consistent across lots the gene expression of excitatory and neuronal genes. And so utilizing this model, we then, to model FTD, engineered into the iPSCs a heterozygous R493X missense mutation into that parent isogenic line, and then ran them through the NGN2 differentiation process, and the resulting excitatory neurons have a haploinsufficiency of granulin. And we've confirmed this by a Western blot, where we see the granulin R493X neurons show reduced expression of the 6 kilodalton granulin monomer, which are produced as a cleaved product from the progranulin gene. And this is sufficiently decreased compared to the healthy control. And then on top of that, we see that in using this differentiation process, we can compare the healthy and the disease model and look at purity of the product, looking at on the right-hand side at beta-3 tubulin, and see that the differentiation is consistent both across our healthy line and our disease line. So the starting material is coming out of the gates very similar, with the exception of the R493X granulin mutation in the disease model. So together with the Maxwell team, we use these neurons on the 24-well HD-MEA plates to longitudinally characterize the neurons. And I will add at this point that this was a nice collaboration between Maxwell and the FCDI application team, in that experiments were run in both locations following similar protocols and were highly repeatable between the two. So I'll introduce the protocol, but then the data has been collected into one set of data because of that consistency. But that was just a nice feature to add, speaking towards the consistency of the Maxwell platform and the cells in combination. And so back to the protocol here. So to do this setup, we co-cultured healthy and diseased neurons with our iPSC-derived astrocytes, so iCell astrocytes 2.0, and then plating about 385,000 cells per well at a 5:1 neuron to astrocyte ratio. And then we looked at the longitudinal development of the activity, so every 3 or 4 days, recording activity post-plating. And so we can look at some of that data set here. To start, we can compare the representative firing rates and active area maps at 3 different days in vitro. That's what's shown on this slide. Apparently healthy normal in the column on the left, disease on the column on the right. And as early as day 15 in culture, you can already start to appreciate an emerging difference in the mean firing rate and overall active area in the cultures of the granulin model. And by 29 days in vitro, the granulin model clearly shows more overall activity and higher mean firing rates compared to the healthy control. And we could take these data from those raw plots and extract them and analyze them across a number of parameters, including firing rate, active area, spike amplitude, interspike interval, just to name a few that are shown on the screen here. And look more holistically at the trends over time longitudinally between the models. And so we can see here that the mean firing rate and active area between healthy and granulin neurons can be seen differences on the top 2 plots. And then while we look at the bottom, the interspike interval and the spike amplitude, we don't see much of a difference between the GRN, aside from a small deviation in interspike interval during the early phases of development that sort of evens out at the end. Another way to look at this that is more holistically is by performing a principal component analysis, which is shown on the right. This is of the burst metrics between the healthy and the disease line, and takes into account multiple spike parameters. Each dot in this plot represents individual bursts in the different respective lines. And using this method, you can clearly see a clear separation of spike burst characteristics, distinguishing the healthy disease line from the disease line. All right, and then another nice feature of the Maxwell platform is the axon tracking software. And we really want to take advantage of this because it was a very unique application offered by the Maxwell system. And this allows for extraction of single cell spiking patterns and enabling the tracking of axon potentials along single neurons. And so on the left-hand side, hopefully the video is displaying an example time lapse shown here of a single neuron and tracking the activity of a firing event in that neuron. With the red lines showing the propagation of action potentials and tracking that propagation down a neurite of an I-cell-induced excitatory neuron. And so we've used this technology to track neurite or activity development over time. And so on the right-hand side are 3 individual snapshots at different time points of the same neuron, looking at the development of the action potential from the same neuron from day 28 through day 42. And you can see and appreciate that the red lines show that between day 28 and day 42, an elaboration of the axon propagation within that neurite, and probably likely indicating an expansion of the neurite length, but also the extension of the ability of the neuron to propagate the action potential throughout that neurite. So that can be, it's a very exciting feature, but it can also be quite informative. And so, for example, in evaluating disease phenotypes here, on the left are representative individual footprints selected from both the healthy normal and the frontotemporal dementia model we're just looking at day 50 in culture. And we show the images show the spatial distribution of action potentials across different neurons. And you can see that we see between the healthy neuron and the GRN of truncated neurite or axon propagation in the frontotemporal dementia model, and that this was corroborated at FCDI using classical neurite outgrowth assays on the INCU site, where we also saw lagging neurite outgrowth kinetics in the frontotemporal dementia model. Suggesting that while the neural activity may be higher that we saw in the MEA activity, the cells may have more immature network connectivity or synaptic development. And so to add to that, at the single cell level in a separate collaboration with Sophion and using their single cell patch clamp technology, we could also compare the GRN model to the healthy, doing single cell electrophysiology, and this data was corroborated. This corroborates this hypothesis or follows this hypothesis. So in this, we've recorded action potentials in the two different disease line, the disease line and the healthy line, and classified the action potentials into 4 different types, which are listed across the top of the screen, where one type is inability to fire action potentials, and the far right is the multi-train action potential capacity. And so below that, we can classify wild type healthy neurons to the GRN and evaluate the different types of action potential characteristics individual neurons have. And we can see that in the GRN line, the classification skews to more the type 2 and type 1 compared to the healthy that has more type 3 and type 4. Indicating that the GRN has more immature action potential capacity and maybe a more immature phenotype, similar to what we were seeing on the MaxTwo platform. And so what does this all mean functionally? Well, we're really in early days still for understanding how these data correlate to dementia. But really the HD-MEA longitudinal data here provides us a window to be able to start interrogating this and asking more specific questions in that vein. But generally, it's looking like the possibility that the difference we're seeing may be causing an alteration in network maturity in the FTD model that is impacting key neuronal processes, potentially excitatory and inhibitory balance, long-term potentiation capacity, and all of these things collectively could be contributing to some of the dementia and the neurodegeneration that is observed in the clinical disease. But again, early days, lots of work to make that connection. All right, so moving on, I wanted to quickly run through some data we've generated with our other neurodegenerative disease models. So the first continues on with a different model for ALS FTD, that spectrum, using our eye-cell motor neurons. Starting off with just some general characteristics of our motor neurons. Here, these are highly pure, as you saw at the beginning of the talk, and a very robust differentiation process. And we've expanded from healthy neurons to also include disease models for ALS, and particularly focusing here on TDP43 mutants, which were engineered into our eye-cell motor neurons. All right, and so TDP43 is a crucial protein involved in RNA processing and is predominantly located in the nucleus of the cells. Within the context of ALS, TDP-43 undergoes pathological changes, leading to its mislocalization from the nucleus into the cytoplasm, and there it forms toxic aggregates. And these aggregates are a hallmark of ALS and are found in nearly all cases. And this has been recapitulated in our cell motor neurons, where we see in this example generated by our CRO partner Axum, that TDP-43 staining in the TDP-43 mutants shows a more cytoplasmic staining compared to the healthy controls. If you look at the top two columns, those images you can see on the right-hand side, a more cytoplasmic localization of the pan-TDP-43 antibody staining. This mislocalization can be exacerbated or induced further in both models using stressors such as sodium arsenite in this case, and then be visualized again using a pan-TDP-43 marker as well as a phospho-TDP-43 antibody. So as we do with many of our disease models, we also wanted to see if we could detect neural activity differences between them. And Pravina already showed a little bit of this data at the front end of the talk here. Again, this is using the MaxTwo 24-well plate system. And what I think we can appreciate from this is that within one plate, we can look at the healthy and the two disease models here and clearly see a difference in neural activity firing patterns between the three conditions, especially compared to the healthy control. But then importantly, as you go down the columns, these are individual wells and replicates of the same cells on the same plate, and seeing a very consistent phenotype across in the healthy and the disease lines, speaking to the reproducibility again, and the high throughput nature and benefits of the Maxwell 24-well plate. And so having this multi-well capacity was really helpful in us building confidence in our cell modeling and comparing across disease lines. So again, this is just the tip of the iceberg in understanding the connection of TDP-43 mutation and mislocalization and the impacts on the relationship with neural activity and furthermore disease progression. There is certainly some literature out there that suggests TDP-43 function can impact synapse function and general neural bioenergetic handling within a cell. We've done some analysis looking at RNA-seq data, and we do see aberrant expression of different pathway-specific genes, calling out on the bottom right-hand corner here, reductions in synaptic protein expression, specifically in the mutant models compared to the healthy normal models, where we see some synaptic genes like PSD95, synapsin, and ACAP reduced in the mutant models, suggesting potentially alterations in synapse formation in those disease models. Again, lots of work going forward, but an interesting pathway to follow up and trying to make that connection between MEA, RNA expression, and TDP-43 within the clinical disease. All right, and then last, a small section here on Parkinson's disease modeling using HD-MEA. So again, just starting off, this is just a slide showing our collection of off-the-shelf disease models within Parkinson's. Again, this is a mixture of healthy lines. And our A53 model was engineered to express this synuclein A53 mutation. And then a combination of the other cell lines being derived from Parkinson's patients containing risk-associated mutations in GBA and LRRK2. And so with this, we can create a nice panel of Parkinson's mutations. And the one I didn't mention that is shown on this screen is, and I'll mention a little bit further later, is the newest addition, which is the TNM175 lines. But what this slide is supposed to exemplify is the ability of trying to set up a panel of assays so that you can compare across disease lines and healthy neurons together in things, high throughput assays such as the HD-MEA. All right, and some example data here is shown using our the A53T, the GBA, and the LRRK2 lines. One indication of a healthy dopaminergic neuron is the development of activity. So we're really excited to see that all of these neurons with the different mutations do develop neural activity over time. And you can start to appreciate potentially some differences in the synchronized bursting patterns and the overall activity levels across the lines. But if we look a little bit further and a little more in depth outside of those raster plots and look at branch level plots to give us a deeper analysis, we wanted to look using the axon tracking and action potential propagation modules within the MaxTwo platform to look at conduction velocity across the different cell lines. And so this software was really useful because it tracks the branching of neurons and calculates peak time differences and cumulative distances across electrodes to estimate velocities of the axon potential propagation. And so using this analysis, we were able to see differences across the disease models, with the GBA action potential conduction velocity being on the high end and almost twofold over what we were seeing on average for the healthy dopaminergic neurons. And similar data was published in 2021 using icell dopa neurons, where they saw similar phenotypes, just specifically comparing the A53T versus the apparently healthy normal. So again, robustness of the cell types and the phenotypes across different groups, seeing the same things. So very, very helpful to see that recapitulation. All right, and then last, so if you work on neurodegenerative diseases, you're likely familiar with TMEM175 as an emerging target, an exciting target for treating Parkinson's disease. So TMEM175, as a bit of background, is a lysosomal cation channel that can control flux of both potassium ions and protons, and it's essential for controlling the lysosomal pH and homeostasis of that. And targeting this channel this channel has been targeted as a therapeutic target because of the ability to control and modulate that pH, because pH changes in pH can impact protein degradation and is associated with aggregation and neurodegeneration in neurons. So genome-wide association studies have revealed numerous mutations within the TMEM175 gene that result in that can be associated with Parkinson's disease. So most kind of the high profile one is this M393T as a risk factor for Parkinson's disease. And then as the flip side, there's the Q65P variant that is considered a gain of function mutation and protective. So these mutations alter TMEM activity, impacting lysosomal pH, and with the decreased activity being affiliated with Parkinson's disease and increased activity being affiliated with protection. So we haven't done this yet, but similarly to our GBA, LRRK2, and A53 dopa neurons, we plan to use the MaxTwo system to profile our panel of disease lines within the TMEM family to see how that relates to overall neural network development and activity. Again, trying to make that link between activity and genetic mutations. But just before we leave here, just showing you some data that we collected, verifying the phenotypes of the mutations in our lines. So this data was also collected by our CRO partner, Axum, utilizing their experience in the technique of lysosomal patch clamp. So briefly, what they're doing here is they're culturing icell dopa neurons, healthy disease types, and then treating them with vacuolin-1 to enlarge the lysosomes, and then they can patch into those lysosomes and record current density plots from them. So they've done this in both control conditions, but then also using a compound called DCPIB, which is a known activator of TMEM175, and they construct current voltage curves and then look at the differences between the disease lines. And so on the right-hand side, we can see that this DCPIB compound binds to the channels should activate TMEM175. It binds equally between the disease lines. But then if we look at the current density plots, we see that a range of differences between the healthy and the different disease mutations, with, again, calling out Q65P here in orange with a higher current density as expected, as that mutation is supposed to increase the conductance of the channel, and therefore we're seeing that as well. So again, sort of confirming at the baseline the phenotype of these cells. Again, we want to put these on the HD-MEA platform to get a better understanding of their network capacity. All right, and with that, I'll conclude here just with a couple statements. Just hopefully I illustrated to you at this point that our FUJIFILM Cellular Dynamics has a wide range of iPSC-derived neurons that can be useful and used on the HD-MEA platform. And we can use these models in our disease models to look at disease phenotypes. And then we were really just really excited about the 24-well plate system because of the ability to create these panels of healthy and disease lines, run them in parallel and in replicates at the same time, which creates a lot of power and efficiency to our system. And with that, I'll just say thank you and hand it back to Marie.
We are always very happy here at Muscle Biosystems to, partner, collaborate, and work together with you there at FCDI. And of course, we are also very happy to see the results today, where we can have a good match, a great match, focusing and highlighting the consistency of cell lines at FCDI, and also the high reproducibility of the activity from these cells using the MaxTwo. So thank you so much for all of these results. I would like to share a few announcements. So first, can you see my screen? All right. So we would like to meet you if you are in the area in 3 different events in the upcoming weeks. So we'll be in New York for the Cold Spring Harbor Bioengineered Tissue Systems and Models, in Tampere University for the Finnish Organ on Chip meeting, and also in Belgium for this New Horizons in Neurodegeneration meeting at VIB. Also, any one of you joining SFN this year, we have some events around that time. So first, we will have our second microelectrode arrays in your science social, wherein we'll have this is a satellite event at SFN. So you have a link that will be appearing in the chat very soon to see more information about this. And if you are joining, we also have a travel grant for SFN. If you're a young researcher, please take a look and apply for that. You can win up to 2,000 Swiss francs for your flights and all of the expenses. And finally, we also have an art contest called the Masterpiece Art Contest. And if you have any works with MEAs or a drawing inspired by MEAs, please submit them by October 17th, and you will get fantastic prizes during SFN. So that's it. Now let's go to the questions. All right. So first we have a question from Jakob. Hello, Jakob. Nice to see you. Greetings as well from Basel. He said, greetings from Basel. We are in Zurich, so greetings. So he has a question regarding the use of I-cell astrocytes. How critical is it to pair diseased astrocytes with diseased neurons to create an effective in vitro model? So he mentioned a paper by Lee et al. 2023 alluded to this being important in the context of GRNR49-3X. For example, I think that is slide 16 to 18. Furthermore, does Fujifilm plan to develop and offer lines of diseased astrocytes? Scott, I think this is for you.
Sure. Yeah. Thanks for your question, Jacob. Yeah, that was a good observation. That Lee paper does allude to an importance of the astrocytes with the heterozygous R493X mutation. So, that's one thing I think that's nice about using iPSC lines is the ability to mix and match and study the combination of potentially study the combination of mutations existing in astrocytes and neurons together, astrocytes on their own, neurons on their own. So I think that combination or that matrix would really help understand the contribution of each cell type for the disease or the cumulative contribution together with them. So I think that's great. And I think that, it's just depending on the question you ask is how in terms of criticalness it depends on the question that you ask. But we do encourage having astrocytes in the cultures at all times, because that really does help advance the development of network activity. And we feel like it recapitulates that environment more truly. And then in terms of your second part of the question, offering disease lines in astrocytes, yeah, we're exploring that option. We don't currently have any available for that. One thing that we have done in the interim is try to do like lentiviral or viral introduction of genetic mutations into the astrocytes, and then co-culture them with you know, an engineered line of neurons together to try and put that duality into a dish with our astrocytes. So that's one technique that could be used in the short term. But right now, just we don't actually have disease phenotypes with our iPS astrocytes available.
Thanks so much, Scott. And thank you for the question, Jakub. Next question we have from Robert. Thank you, Robert. He said, thank you for your talk, Scott. So do you generally see an increase in synchrony and decrease in overall firing in glutamatergic neuron astrocyte co-cultures relative to the neuronal monocultures? And is it still possible to perform axon tracking in the presence of astrocytes? Maybe that part we can answer on our side as well, but you can also do so. Or is there some level of physical occlusion by astrocytes that prevents clear readouts? Maybe we can jointly answer these, Scott and Pravina.
Yeah. Yeah, so I can step in first. Yeah, so of course, astrocytes actually enhance network synchronization by modulating extracellular ion concentrations and releasing gliotransmitters, essentially. So it's actually very important to include them, as Scott previously mentioned, in a co-culture with neurons. We actually can record axon tracking. So the axon tracking data that you saw is with both neurons and astrocytes co-cultures. So it does not mask the signals of neuron action potentials.
Scott, would you like to add something? Yes.
No, I agree with that. What you said. Yeah, I mean, we haven't had any issue recording and identifying that activity to a neuron and action potentials. So it should be no issue.
Thanks. And I'd like to add something to it. So as Pravina already mentioned, I think the astrocytes allow us to even better record the axons, just because the neurons become more active. And with this, even though there's a layer of astrocytes on top of the electrode, because they're very, very sensitive, they can be detected even if the neurons are a little bit on top of them. So yes, it's possible. Thank you so much for the question. Next is from Max. Hello, Max. What have been some insights from these setups? I think the combination of FCDI cells and MaxTwo HD-MEAs for Alzheimer's disease. Has there been any experiments done so far? Not that off, but
yeah, maybe I can speak a little bit from our side of things. We have done a little bit of data we have some Alzheimer's disease models, looking at like the different APO isoforms and things, mostly looking at like using calcium in fluctuations to observe activity. So less so on the MEA platform, although we have done some initial work on that, and at least in the calcium assays that I can speak to, we do see differences in oscillation patterns. Those are a surrogate for neural activity, the calcium oscillations. So I would anticipate on like HD-MEA platform to also be able to see some differences there. But I don't know if Pravina, you have any additional background from maybe other users who have tried this and are modeling Alzheimer's.
So of course we know a lot of users who are working in this field. So there's data that our users can present, but yeah, we have not tried it in-house yet.
Yeah,
but of course it's of interest.
So Max, perhaps you can, I don't know, forward you some papers, if so, or you can take a look at our resources page and you can find them there. Yes. Thank you so much. Bo, nice to see you here in our webinar. I think this was already answered before, but let's ask it again, Scott. Do you always use wild-type astrocytes in these co-cultures?
Yeah, and the data you saw, there was always wild-type astrocytes in those cultures.
Yeah, thanks a lot. Next question from Janos. Hello. With the IPS route, you rewind the clock to 0. And then the dementia model, the disease only manifests in 60 plus years, but the experiments are only after 2 months of differentiation. So why do you expect any difference with the mutations? I guess this is more of a IPS kind of disease modeling question. Scott, would you like to answer?
Sure. Yeah. That's an insightful question and something that we're always thinking about at FCDI in terms of disease modeling. We know that the engineered mutations that we put in to model these diseases or the patient lines are robust mutations, and part of the reason why we put them on these MEA platforms is to see if we can see phenotypes. And in a lot of cases, it can take time for those things to manifest. The data that we showed today, with the GRN model, it was interesting to see that come out so early in the cultures. But I would imagine in other models, time is going to make a bigger difference to look at, dementia models, particularly in Alzheimer's disease and some of these longevity things. But I think those questions can be asked using this combination of cells in a platform because of the ability to look longitudinally. So I don't know if I have a great answer for you on that, except that the cells express the mutation we're trying to profile just to see if they're there. They are, and that's where we try to ask more questions about why, how, and how it relates to the disease. But if you wanted to do long-term longitudinal studies, I think the cultures can be maintained for quite some time to actually test that thought.
Thanks a lot, Scott. Next question we have from Bien. I hope I pronounced that properly. I think this is for Pravina. For 24 well plate, is it possible to assay different samples probably on the same plate, such as iPSC-derived neurons and organoid at the same time?
Yes, so of course you can try multiple different cell lines or multiple different organoid lines in one plate. And if you actually wanted to mix iPSC-derived neurons and organoids on the same plate, this is possible. Essentially, our plate, our MaxLab Live will record from all the wells, and so you could have a particular row for iPSC-derived neurons, and then do the plating later for your organoids on another row and record from them all at the same time.
Yes, I guess now it just depends on your
use case.
Your use case. If it's easy for you to plate all of them on one plate, like different samples, then of course that's possible. Or you can just culture the same types also with the different types of lines. All right. Next we have Jakob again. Hello. So this Scott, I think this is for you. Regarding the motor neurons with TDP43 mutations, he's curious about their functional characterization. Do you see a change in the observed differences over time in your longitudinal cultures, and how long did you try to culture them?
Yeah, we typically culture we start recording around day 7, and we'll culture and terminate anywhere between day 21 and 35, 50. Just it depends kind of on our abilities at the time and the depth of the question we're asking. So the data that we're showing up on the screen there, I think it was from day 14 in culture, if I'm remembering correctly. And then I know we have, I'm just trying to recall if that changes over time. I think the disease lines that the phenotype maintains itself over time. And I think we went out till about day 35 with those. But there's an application data set that we can share with you after this, Jakob, that would give you maybe some more insights into your question.
Thanks so much. I'll now jump to the question by Arms Zawa. I think this is Ropa. So thank you for the great presentation, Scott. So you mentioned that in the beginning about the triculture systems. Could you talk a little bit more about these cultures? And do these cultures mostly consist of neurons, astrocytes, and microglia? What does that media composition look like? And do microglia equally boost the activity development as do the astrocytes?
Yeah, great question. Yeah, so the triculture systems that we have traditionally set up on MEA, when we're referring to triculture, we're thinking about excitatory neurons, inhibitory neurons, and astrocytes together. So modulating neural cells with the astrocytes. And then we do have another triculture mentality where we're adding in microglia to that, where it's neurons, astrocytes, and microglia. And so on the MEA platform, there's specialized media that you use to help that neural network activity develop. In this case, we're using brain fizz to do this. And we know that microglia don't like to be in brain fizz. There's something about the composition and the ionic balance there that the microglia don't like. So we have not integrated microglia into those cultures yet for MEA. But we are exploring like alternate media formulations that we do know, and we have identified some promising candidates to combine all those 3 cell types together and preserve MEA activity so that we could ask that question about how microglia are actually impacting development of activity in relationship to astrocytes and the neurons in the culture. So I think to be continued on that one. Yeah.
Yes. Thank you. Due to kind of time constraints, I think we'll end the Q&A at this time. If you have any questions about FCDI products, please reach out to Scott and the team at FCDI. And if you also have questions about MaxTwo HD-MEAs, please contact Pravina or myself. Just a few words from our side. So thank you so much for joining our webinar and Q&A session for today. Thank you once again, Scott. So we have our postcards around the world. So if you have them, if you took them at the booth, please take a picture and send it to us here at Muscle Biosystems. And also, we have our next webinar upcoming on October 28th. So this is going to be by Dr. Siuchuan Lin and Dr. Philip Hornauer. And please stay tuned for more information. We'll send it via newsletter and also on the website. Thank you once again, and hope to see you in the next webinar. Bye.